a global ambassador for BIO110, the class. So today my topic is prokaryotes. The OG survivors of planet Earth and the lab experiment where we actually transformed bacteria. Prokaryotes are tiny, like really tiny, but don't let their size fool you. They're everywhere, in soil, water, air, even inside our body. They will survive boiling water, heat, frozen, cold, salty lakes, toxic environment, literally places where humans would last 3 seconds. How do they win so hard in life? First of all, they reproduce asexually through binary fission. They split themselves into two. They create another version of themselves. And second of all, their DNA changes to mutation or genetic exchange. So they're adapting to their environment in a lightning speed. They are split into two groups, bacteria and archaea, and they are unicellular, which means single cell does everything, and their cell is much more smaller than eukaryotic cell, which is 0.5 to 5 micromillimeters. Prokaryotes are tiny. And their shape is split into three groups: spear, rod, and spiral. As you can see on the picture, some prokaryotes have slimy capsule made of polysaccharide or protein that helps them to stick to surfaces or hide from the immune system. Many prokaryotes form metabolically inactive in the spears, which can remain viable in harsh conditions for centuries. Some prokaryotes have a hair-like appendage called fimbriae that allows them to stick to their surfaces or other individuals in colony. About half of all prokaryotes exhibit taxis, the ability to move towards or away from the stimulus. For example, chemotaxis move towards nutrients or away from the toxins. Flagella are the most common structures used by prokaryotes for movement. It may be scattered about the surface or concentrated at one or both ends of the cell. A key feature for nearly all prokaryotic cell is the cell wall. It maintains cell shape, protects the cell, and prevents it from bursting in a hypotonic environment. Most bacterial cell walls contain peptidoglycan, a network of sugar polymers cross-linked by polypeptides. Archaea contains polysaccharides and protein but lacks peptidoglycan. Scientists use Gram stain to classify bacteria by cell wall composition. Samples are first stained with crystal violet dye and iodine, then rinsed in alcohol, and finally stained in red dye, which enters the cell and binds to its DNA. Gram-positive bacteria have simpler walls with large amount of peptidoglycan. Gram-negative bacteria have less peptidoglycan and the outer membrane that contains lipopolysaccharides. Therefore, as many antibiotics attack peptidoglycan, gram-positive bacteria are extra vulnerable. Let me introduce you my favorite chip in the world. It's only in China. Oh God. Put it aside because we're learning about bacteria and prokaryotes. So, the prokaryote is used to make antibiotics, insulin, vitamin, and others. They're very critical in food, especially yogurt, cheese, and kimchi. They help producing oxygen, and they clean the spilled oil to bioremediation. And they maintain our gut microbiome, which is very important, and without them, we will not survive for even a week. But they have a harmful impact. Let me read it from the book, our book. Pathogenic bacteria typically cause disease by releasing exotoxins or endotoxins. Horizontal gene transfer can spread genes associated with tolerance to harmless species or stains. Resistance to multiple antibiotics have evolved in many pathogenic bacteria, a problem of great concern for public health. So, So it's so situation, and I'm glad that I didn't eat the chip. Now the fun part, our lab experiment. We used E. coli bacteria and tried to transform them using plasmid called PUC19. A plasmid is like a USB stick for bacteria. Plug it in, and suddenly the cell has new abilities. PUC19 gives bacteria an ampicillin resistance, AMPR gene, and the LACT gene, which turns colonies blue when growth within IPTG and XGAL. We made 2 tubes, one with plasmid, one without it. We kept them both on ice, then gave them quick 45-second heat shock at 42 Celsius to help their DNA enter the cells. After cooling them back on ice, we let the bacteria recover in LB. Then we plated them on non-selective plates and selective ampicillin plates. As expected, on the non-selective plate, both bacteria with or without plasmid grew. They showed a long growth. The color was a little different. One was beige and the other one was white, but they both showed growth. But with the plate with ampicillin, only the plasmid treated bacteria showed growth. The other one didn't grow at all because it was not ampicillin resistant. So this result was pretty popular among my other classmates' result, but our result was had a little variety of differences because of the heat time shocking timing, and how they spread the bacteria sample with glass beads, but it was pretty popular among them, and this plate showed growth because of the LACT gene and ampicillin resistance gene I mentioned earlier. And that's the world of prokaryotes, small, ancient, adaptive, and absolutely essential. And our experiment on them and seeing the transformation that happen in real time. Also, yeah. And thank you for joining me as a BIO110 ambassador, and hope to see you in the future too.